One of these things is not like the others.
An interesting story.
What's that?
I got laser eye surgery several years ago, and they have to peel a little flap and flap it over, and then shoot the laser in your eye. And I sat for an hour afterwards, and then they checked it. And when I left the building, I pushed the door open, and there was like a change in the air pressure or something, and a big gust of wind came, and it flapped the flap open. So I had to go back in and get them to take a look at it. My first thought was, oh, my contact like came dislodged, and it wasn't my contact. It was like my cornea. Or
How about that?
I have to say, you just never know with people, I guess, because I said one of these things is not like the others. And I was talking about Jeff's shitty lighting, and James launched into some kind of diatribe about his LASIK surgery.
Well, Jeff was talking about the glare on his glasses, and I just joined, so I figured you were going, you're talking about my 20/20 eyesight.
No, I wasn't anywhere near your 20/20 eyesight.
Well, in that case, how's my lighting?
You have brilliant. You have the best lighting of all of us. I really.
Yeah, James, we could take a lesson from you.
Now, Andre Domino, I know you've been currying favor, and you want me to come out on camera and say that you're my favorite member of the group, with your sycophantic peddling of my brand over your shoulder. And believe me, I get it, and I love you for it. I do. But isn't that last week's sign?
You're absolutely right. I didn't have a chance to change your sign, so it's still there.
Yeah. I mean,
Well,
when you
it
did
still
the one with
applies.
the Christmas ornament, I was like, wow, this guy's really into it. And now this week it's like, fuck, I don't have time. I mean, where's the sign from last week?
I emailed it to you.
I wasn't addressing myself as honey. I was role playing there, where you played the role and you were addressing your spouse, looking for the.
We'll have a new sign next week. Don't worry, Joe.
You say that. You got to stay on top of this stuff, because I'm bringing Craig Solid on now, and Craig's a new member, and you're on a very precarious perch there. That's all I have to say. You know how capricious parents can be. They love all of their children. Now, in the case of Dwayne, he's like, I have one child. I don't really know what that's all about. If I had a second child, I don't really understand. Would I like it more than the other? Is that really a thing? But how many of us have more than one child? 3 of us. No, okay. Yep. John, who's your favorite child? They're not going to watch this broadcast. The chances of them watching Dad.
What do you know?
Yeah.
They are.
The chances of them tuning into the medical MDG premium broadcast about sterilization in the hopes that Dad is going to not talk,
It's going to be viral.
but then say to the world who his favorite child is so remote. So give us something here. What do you think?
Yeah.
How many children do you have?
Diplomatic, because they're both different.
I know.
It's amazing how different they are.
But which one do you love more?
I'm not answering that.
Terrible.
But there is an answer, right?
Both.
Terrible.
Both is like the lamest and
Of
oh,
course it is. Diplomatic.
Lawrence Stevens, you came into the city for this? You know what I like best about you, Larry, is every week when I address you, you talk into your muted mic. And there's never any kind of learning. There's never any growth.
I guess this is the
mic.
Joe. This is the Zoom roast by Joe H. So
Larry, do you need some help there?
It's pretty complicated. I get confused
I
easily.
know. Superannuation's a bitch. Rob Packard.
Hi there.
Hey. I don't have a joke.
What have you been up to, Joe?
no good.
I cleaned my office a bit.
Your office is always nice and neat and clean.
No, that's the nice and neat and clean part that I allow you to see.
Well,
The New York
The
plate.
detritus that is right underneath the.
yep.
I'm actually floating in garbage.
What's the New York plate, though, Joe?
That's my father's old license plate. That was on his Cadillac. Daddy liked caddies.
That's great.
Mm-hmm.
That is cool.
Mm-hmm. Maya's joined us. I told her all about our little family. Josue, we can see. He joined us today. That's nice. Shepherd in the house. There are no women on the screen. Ah, here comes one woman. Hurry up, Maya. We're waiting on you. All of the men are waiting on you.
It's not a dating app, Joe. It's a Zoom.
It's not a dating app, but still, I mean, I wanna show
Swipe right,
diversity.
swipe right.
And I've just got a whole bunch of white men.
How about that?
It's kind of like the last administration, right, Larry?
Yeah, yeah, what can you say? Life goes on.
It does.
Joe, you're on fire this morning.
Maya, for the people at home, how do you pronounce all of your names?
Well, in English, I would say Maya Grinstead Hilliges.
Yes, but in your proper native tongue?
Maya Grinstead Hilliges.
Call her
Good mix
Maya,
of-
people.
Yeah.
What would you like the nice people at home to know about you?
Ooh, let's see. Well, I'm from Denmark, hence the funny pronunciation of part of my name, but live in Wisconsin. And I work for my dad's company, Paramedic Lasers, and we produce and develop laser therapy equipment.
Used by whom?
Sorry?
Used by whom?
Chiropractors, physical therapists, acupuncturists, massage therapists, veterinarians, some at-home use as well, MDs, so kind of a plethora of practitioners.
I totally would have thought dermatology when you said laser.
Yeah, some dermatologists, but it's more, so the lasers that we make are more for accelerating the natural healing. So you'd like a sports injury, like a sprained ankle, for instance, or if it's tennis elbow or carpal tunnel syndrome, back pain, some skin issues as well, like wounds or acne and things like that. But just in general, it helps the body heal itself faster. So increases the blood circulation, increases the production of ATP, so more energy for the cells, reduces inflammation, reduces pain. So really, in short, just helps the body heal itself faster.
A very engaging young woman, ladies and gentlemen, and she doesn't know it yet, but I'm going to ask her to speak in March, and we're all going to give her some concentrated consulting on how she can expand her footprint in the United States.
Sounds good.
Are you going to discuss how the technology works too in March? This is great. I'm interested in this conversation.
Sure. I just learned this just now that I've been speaking, so sure, I can get into that.
Yeah, I'm going to bring my wife along. She's a physical therapist.
Oh, sure. Yeah. Awesome.
Yeah.
Maya, right now I'm telling you, if you sell a unit to the Mancinis, I am totally getting a 10% commission. Everyone here heard that. I don't know what your margins are, but there's got to be some kind of rules here.
I'll keep that in mind.
Oh, Sway, we've not seen
Hello, guys.
for a long time. Where have you been?
Been super busy. This year started super busy. So I haven't had time to log in with you guys, but making some time today.
Appreciate that. You know, I feel as lucky as my mother when I call her every 3 months.
You should definitely call her more often than that.
There's a parallel.
As I should call
She
more
knew-
often every
This
week.
is the point I'm making, Josue.
Yep. Yeah,
We make time for the people that we love.
definitely. No, it's great to be back with you guys. And I was listening to the earlier intro and just remember how much I miss being here on Friday morning.
I forgive you now.
Cheers.
Apparently Michelle Lott is not Michelle Lott. This is Diana Benson, who decided to log in under the guise of Michelle Lott, but she too is allowed to unmute herself and put on her camera. As she too is.
Is that a hint, Joe?
Yay.
Turn on my camera.
Hi. Tell us about your mug this morning.
Good morning. I do have a lot of mugs that are very large, so gotta hold a lot of coffee.
They're all caffeinated.
Yeah, they all have cute little things on them.
I'm going to publish a video that I shot with Ty and with Michelle earlier this week, next week. Give the kind people at home just an indication what we talked about real quick.
So on Monday, the FDA published for public comment a list of devices that they are proposing to down classify. They are proposing to down classify about 83 devices from class 2 510K, and they are making some of them 510K exempt. And the devices on that list are quite shocking, actually. If you haven't take a look at the 35-page summary,
Which you were
really
going to send me and failed to do. Go on.
Oh, sorry, Joe. I'll follow up on that.
Mhm.
Definitely go read and take a look, because things like ventilators for home use are on there. Antiviral surgical masks are on there. It's very shocking what the criteria were. So Michelle
And you
and I
speculate the reason they did this was,
actually, Michelle and I just had a conversation about that, which you would be very happy to know we recorded, because she figured it out. So that
and that
might
is,
so the device classification, I feel like I'm stealing the show here.
you are, Jeff, we don't have time for your talk today.
Okay.
And all you people who've chosen to shut off your video cameras, what are we doing here? Come on, say hello to the nice people.
So what Michelle and I talked about, she's the genius behind this, and she talked to some other consultants, is that there were basically 3 criteria that the FDA used to publish this list. And it was definitely based on some sort of algorithm that we feel like didn't have human review. And so they looked at the number of submissions, they looked at the MOD database before the EUA and after the EUA. So they looked at the number of adverse events that had happened before and after, and then they looked at the type of devices. And so some of these high-risk devices that are on here, like antiviral surgical masks, there's no reportable events because the total amount of devices ever cleared was 3. So of course there's no adverse events, because 2 of them aren't even you can't even purchase them anymore. They're not manufactured. And you can't get the volume that the algorithm was looking for because they're not available, because the testing requirements were already so strict. So manufacturers didn't think that the burden for the antiviral claim was worth it necessarily. But now, because that list wasn't screened, it pops up because they're not manufactured. There's no mod events, so therefore they are a low risk device because they haven't seen anything reportable. When in reality, it's that the testing criteria was strict enough that it had appropriate risk with it. And instead of just reevaluating the criteria needed to make those claims, they just made it 510K exempt, which means any manufacturer can produce it and claim that they're manufacturing it to the level of controls. And they can distribute for 5 years before the FDA ever thinks to do an inspection.
So it sounds to me as though, because I, as far as I know, have no criminal record of manslaughter, I should pretty much be able to just go and kill people for a while before they catch on and realize they need to regulate my ability to kill people.
Well, I think, I'm an optimist, and I feel like you have to be in medical devices. It's more that I think people don't know that they're not meeting the requirements or the criteria. And a good example of that is the recent increase of testing burden for just plain surgical masks that the FDA has come out with. They've really increased their scrutiny of the AQL 4.0 for devices. Before it used to be, you would test 50 to 60 masks, and now they're asking for 3 non-consecutive lots at this AQL level. The magic number that people are saying is like 32 per lot, so 96 per each of those 5 tests. And before, you could test like 10 masks. And so this list and the FDA's testing requirements aren't in alignment. And this list is out for public comment, so take a look for yourself. And
When she
Share
shares
that
the
list. Can you
with
share
us.
the list?
yeah, hold on.
Larry, you have anything on that before we switch to today's topic?
Oh, I'm not even up on that one. I'm gonna have to look into it myself.
I think the list is very weighted towards products that don't have a lot of sales. Like she said with the antiviral masks, if there's only 2 or 3 510Ks, then there's not going to be a lot of adverse events, because we have another client with a radiologic imaging system that is on the list, and they
Yep.
don't want it to be on the list.
And that's the other thing is that, you know, for manufacturers, they don't want it. They don't want these high risk devices on the list, because if they have it cleared, then the barrier to entry is low. So yeah, it is exactly as Rob said. It is that there's just not a lot of people producing these devices.
Yeah. But this is what happens when you double the staff at the FDA and have people with 18 months of experience supervising somebody with 6 weeks.
I think the cynicism part of the call is going to draw to a close now as we talk about the joys of sterilization. But before we do that, Coop, we've not seen you in a while. You want to say hello to the kind people?
Hi, kind
Yes.
people.
Any news by you?
Um,
Don't give us the whole "we've been really busy" nonsense. I swear I already tried that.
I can't think of anything.
Boring. Okay. Bye, Alan. Sukar, any updates on your device development?
Yeah, hi everybody. We have signed a contract with Galero LLC. They're located in the Research Triangle area, and we have begun production for a full
Nice.
510K Class 2 approval.
Are you working with Tim directly?
Tim Hopper. He actually no longer. I was working with Tim for many years. He
Do you know
left
who that was?
Galero a couple years ago.
Oh,
His son was in a terrible
I know.
accident.
I wrote a post called "He Broke His Neck" years ago.
Yeah, there's a really good YouTube video about it. And he, so he left and he's working for a company, probably in artificial intelligence or something that works with people that don't have limbs and with spinal cord injuries. So I'm working with his predecessor, Mike Chambers, and I'm actively seeking angel investment for seed funding. So I'm close. We're 61 weeks away from FDA submission.
Who are you using to submit? Have you checked out?
Galero has everything. They're an all-in-one stop and shop. They have FDA, they have ISO certified, they can manufacture and distribute 10,000 units in one day. And
I
so
want you to introduce me to Mike, because he should be using Sumya's S&B product. Were you on the call for that?
I don't think so.
He
But I'll
has
be glad to introduce you to Mike.
he has regulatory software that streamlines submissions and regulatory life cycle.
He's actually the engineer. The other guy, Don. I'm working with 4 or 5 different people over there right now.
Well, find me the right one. Let's
Well,
make
yeah.
a love connection.
And we're talking to, I call them the BNCs, the big needle companies, about licensing and acquisitions, and they want to put their little spin on things. So they're going to help some of the financials with Cartech, but I don't want to give them too much, you know.
Do you is your single use, or does your product need to be sterilized?
It needs to be sterilized.
Oh, Jeff Sauter has a thing or two about that. Is that why you tuned in today, or because just general love?
We have about 9 or 10 different ISO sterilization, ISO sterilization techniques that we need to perform for FDA approval. So,
all right, Jeff, our guest today knows a thing or two about that. Mr. Sauter, I realize that the only way to get you on the phone or to interact with me on Premium is to give you an international stage. So here it is.
Well, thank you very much. Hello, everybody. And Joe, much appreciated and this opportunity to present. And so why don't we go to it then? We will start here. Can everybody see the screen?
We can. Why terminal sterilization?
Well, we don't want to exactly start there. We want to start here.
Okay.
Super. So yeah, so basically, I'm going to address the trends in terminal sterilization and the emergence of E-beam and X-ray. Within the terminal sterilization industry, in the last 10 years, a lot of big things have happened that have changed the way that medical device companies, innovators look at sterilization. So we will get into this. And so basically, let's start why terminal sterilization. Terminal sterilization is defined as the process whereby the product is sterilized within a sterile barrier system or final container. So as opposed to aseptic processing or sterilization filtration, where it's microbial exclusion, whether it's any kind of terminal sterilization method, it's microbial inactivation. It's considered the final step in the manufacturing process, but it's absolutely vital to consider your terminal sterilization modality early on in the product development stage. Absolutely critical. Companies have gone down the wrong course, not having taken this consideration early on. With terminal sterilization, whether it's gas or irradiation, product is sterilized to a demonstrable sterilization assurance level, SAL, and it's 10 to the -6. And that means statistically that 1 out of every million devices could potentially still be contaminated. You'll never achieve a 100% sterility assurance level. Very quickly with 10 to the -6. Why 10 to the -6? Well, really, there's no empirical studies. It's really just was identified as the goal to achieve, and it's become the sterility assurance level. There are discussions within sterilization industry with AMI groups with lowering the 10 to the -6 to -10 to the -5, 10 to the -4, and 10 to the -3. And statistically, there's a study by Byron Lambert, who's basically a guru when it comes to any kind of sterilization, but especially with the radiation sterilization, that with 10 to the -3, for the safety of the patient, of course, it's to keep the patient safe, that statistically there's no difference between 10 to the -3 and 10 to the -6. So there are discussions afoot there, but that's a long way off. Right now, it's 10 to the -6. And very importantly, because with the medical devices, there's the merging of drugs, therapeutics, and biologics with combination devices, it's very important to note that with terminal sterilization within the pharma world, they're moving towards terminal sterilization versus aseptic processing and microfiltration. And it's being pushed heavily by both the EU and FDA for pharma companies to demonstrate that their product cannot be terminally sterilized. Otherwise, they want the product to be terminally sterilized.
Hey, Jeff,
Yes.
I want you to back up a bit because I don't know anything about this topic at all. And you've launched, I mean, I understand in general, sterilization, make clean, and you're telling me that I can make clean while it's inside the package in which it's shipped. That's what terminal is, yes?
Yes, indeed, that is the case.
So basically, it's in its package and it's not sterile, and then I can process it and sterilize the thing inside the box. Is that what you're saying?
Indeed, that is the case.
Okay.
With the individual devices within their pouches, and then inside shelf boxes, then inside cartons or shipping boxes.
That sounds like a good idea. It sounds like a more expensive idea than the way things are typically sterilized.
Well, I mean, in all cases, for terminal sterilization, product is sterilized in its final barrier package. Now versus aseptic processing and microfiltration, it's much less expensive. It's a much more controlled process, and it's required by the FDA. I mean,
okay, well, the requirement part makes the other two things novel.
Yeah. So indeed,
but only some things are. So should I think that those things affected by that are required to be terminally sterilized, which is, I'm thinking, the most stringent?
Yeah, essentially. Yeah, essentially any class 3 device is required to be terminally sterilized.
Okay.
Many class 2 devices and a lot of medical devices, to be able to provide sterility assurance, they do validate their products to whatever sterilization method. Even though their product is not required to be terminally sterilized, it is. And we see that even extending outside of the medical devices, especially like with laboratory consumables and disposables. That's moving towards a more regimented sterilization validation program.
You mentioned 3 types of sterilization. How many types are there? Terminal being one.
Well, terminal sterilization, well, there's many types of sterilization, like even within hospitals, they have to reprocess endoscopy systems and surgical.
Right, of course. That's already out of the package and I'm reusing
Yeah,
it.
so that's,
But as it relates to your world of,
and within our world, my world, for terminal sterilization, for the terminal sterilization modalities, you come down basically to 2 types of sterilization, and we'll get into it here. And sorry about scribbling the screens. I can't use my page down. So why don't we go here?
Yeah, you're on
So,
page.
yeah, so basically, the FDA considers there to be 2 categories of terminal sterilization: established and novel terminal sterilization methods. So there's category A and category B. And for the most part, over 95% of all medical devices are sterilized under category A, terminal sterilization methods. Now we have other interesting technologies developing. There's also established category B, which is hydrogen peroxide, ozone, and other novel sterilization methods that are looking to take hold, such as VPA, vaporized hydrogen peroxide, nitrogen dioxide, vaporized hydrogen peroxide.
Jeff,
That
this is what I'm hearing, and I'm really playing the role, which I was meant to play, of guy who knows nothing about this. I'm interpreting sterilization as a concept and A and B as the chemicals we use to do it. Is that correct?
Not the chemicals, but the modalities to achieve that. And really what you come down to is you have there's autoclave systems, you have dry heat and steam. But really when you come to terminal sterilization, you're looking at either gas, and within that realm of gas sterilization, it's 99% probably is ethylene oxide. That's the go-to gas sterilization modality. And then you have irradiation sterilization, which we will get into with the next page.
Okay.
So over 90% of all medical devices, they're sterilized by basically, at this point of time, 3 separate methods. We have ethylene oxide, we have gamma, and we have E-beam. And now big time emerging is X-ray sterilization. You hear a lot about it. It's available in Europe, but not yet here in the United States. And we'll get a little bit more into that. Now, within the gas realm, as I mentioned, it's really, when you're sterilizing with gas, typically you're saying you sterilize with ethylene oxide. And it's the most widely used. And all of these terminal sterilization methods, they're the best terminal sterilization method depending on the type of product to be sterilized. So like with ethylene oxide, if you have active electronic components or certain fluoropolymers such as Teflon, PTFE, or acetal, then you want to look at ethylene oxide because it's not compatible with irradiation. And then within the realm of irradiation sterilization, gamma is the most commonly used form of sterilization. Now with ethylene oxide and gamma, we got some really big problems here, and I'll get into that. And then now we have the emergence of E-beam and X-ray, and especially with sensitive materials and combination products that contain drugs or biologic components. E-beam, because of its flexibility to implement advanced methods, is the best way to go. And X-ray is, along with E-beam, it's the future of irradiation, with availability starting in this year, 2021, and we'll get into that. Now, you may ask the question with ethylene oxide and gamma, why does everybody use ethylene oxide and gamma and not really E-beam and X-ray? Ethylene oxide, probably 55% of all of the medical devices are sterilized with ethylene oxide. Maybe 30% or so is sterilized with gamma.
I'm going to guess because it's either cheaper or that's what all the old systems were made out of.
No, well, basically, ethylene oxide is cheap, and it's probably the cheapest for high volumes. But it's basically what happened is with the terminal sterilization, basically the terminal sterilization industry as we know it basically started, I don't know, like the 1940s. In the 1930s, autoclave was first developed. And then I think in the when was it? 1940s, I believe, ethylene oxide was early 1940s. Griffith, which was formed Griffith Labs, then Griffith Microsciences, then that was taken over by Steri-Tek. That was the implementation of ethylene oxide, and gamma was first used. I think in early 1963, it was a Canadian initiative. So that was used to sterilize sutures. And then we also early on in 1956, Johnson & Johnson was the first company to validate and sterilize medical devices to E-beam. And then Ethicon was the first company in the early 1960s to sterilize via X-ray. So basically the reason is really with technology. Back in the 1950s and 1960s, E-beam and X-ray, the technology really wasn't advanced to really for the throughput and the controlled processes to sterilize massive quantities of medical devices as with ethylene oxide and gamma. So ethylene oxide and gamma had an opportunity to take hold in the 50s and the 60s. And there really wasn't into the 2000s where we really seen that the acceleration, the exponential acceleration of technology for E-beam to make that much more reliable, the go-to technology over gamma. And then now we have X-ray. And we'll get into now, X-ray is just on the horizon. It's in Europe, Switzerland, and the Netherlands. Steris has a couple of X-ray facilities. And I'll get into that, but Steri-Tek ourselves, we're getting big time into X-ray itself. So X-ray will probably eventually displace gamma because there's big time problems there. And we will get into this with here. But first of all, just to give you an overview of the industry, within terminal sterilization, really it's a duopoly. There's two companies that globally dominate the terminal sterilization industry. And they're two very good companies. We have Steris. Yeah.
Wait, let me back up again because a lot of the last slide was a blur to me. I'm not asking you to go back.
Okay, let's go
no,
back.
No, don't go back to it. Don't
okay.
go back. Because I just heard the word sterilization a lot and lots of different ways of doing it. And you have a great history of it. If I ever need a historian on sterilization methods, you're my guy. I'm at the point of, I've got a device, I need to be sterilized. I walk into the sterilization store and I'm looking around and I have no idea how many different kinds there are. You've taught me that terminal is a kind and there are 2 or 3 other kinds. How do I know which one I need? You will say, well, it's a function of what FDA regulates. I say, okay. And then once I decide on that, there's lots of different ways I could go about it. How do you decide which one is right for me? I'd be curious to know.
Which one is right? Early on, to test the materials, the components of the device to prove out the suitability of a given sterilization modality.
That makes a lot of sense. As a
Yeah,
function
early
of what
on.
my thing is made out of, some things will work and some things won't.
Right. You could determine up front what is the best terminal sterilization modality, what, depending on the components, if you have active electronic components, integrated circuits, memory as part of your medical device, or fluoropolymers such as Teflon, PTFE, you want to go down the path of ethylene oxide.
So is this basically a list or an algorithm? I tell you the components and materials that I use, and you type into your fancy computer and it says, "Bling, you want this kind of sterilization." Is it that linear?
No, it's not that easy. So, because like even with Teflon,
that's a job security thing, I think.
Yeah, it is.
Saying that. I can replace you with a spreadsheet.
Yeah, you want to contact us to determine that.
La. Okay.
Before we go down to this.
The answer, I get it.
Yes.
We all have our gigs here.
Yes, right. We need to protect that.
Fair
no,
enough.
But seriously, you want to work with whoever your sterilizer is. You want to work with them as your partner. And now with Steris and Sterigenics, these are the 2 800-pound gorillas. They offer all of these sterilization modalities.
How many pounds is your company?
Pardon?
If they're 800 pounds, how heavy is your company?
Well, we're gaining a lot of weight, but I would say we're probably not even 50 pounds yet. But I can tell you, Steris and Sterigenics, they are well aware of us. We're growing very, very fast.
You're eating their lunch, so to speak?
Well, it's friendly competition. But
is it the 3 of you now? Are you like number 3 in the space?
No, I can't say that because there's sterilizers that are only ethylene oxide that are major companies like Cosmet. There's also, now within the United States, insofar as the radiation sterilization, now there's only one other e-beam, pure play e-beam player, and that's e-beam services. They're based out of Cincinnati and also, or in New Jersey and Cincinnati. Very good company. But Steri-Tek, they just acquired the only other independent e-beam sterilization company, Iotron, based out of Canada, and they have a facility in Fort Wayne, Indiana. So,
You guys are privately held?
yes, we are.
When's the last time you acquired something, if ever?
We have not. We're building new facilities. We're greenfielding new facilities. Our next facility is in Dallas. We're in San Francisco Bay Area now. Our next facility is going to be in Dallas.
Do you have
Then we're
any
gonna be
outside the US footprint?
No, but we're looking, we're keeping our eyes open for opportunities right now in Europe.
Okay.
But right now there's too much growth. There within the sterilization industry, there's no capacity. So
Just a matter of time then before one of these big guys buys you out, is what you're telling us.
No, well, just between us and our friendly group.
International audience, go on.
Yeah, I mean, there's interest. However, just to be clear here, I mentioned we're building a brand new facility in the Dallas area, and I'll get into that. But we're building our $30 million facility in Louisville, and it's a tax advantage area whereby all investments we make into this property, it's tax-free as long as the owner of Steri-Tek holds on to this company for a minimum of 10 years to get the tax breaks. So we have no incentive to be acquired. Now down the road, possibly, these are Steris and Steri-Tek are very good companies. They're very smart companies.
Okay.
They're making a lot of acquisitions, not only within the terminal sterilization, but expanding into microbiology.
So we'll look for your buyout in 2031.
Yeah, well, yeah, but the thing is, we're going to be building other facilities that we're going to be in tax zones. So that's going to be going out for a while. So
you own any equity in this company there, Jeff?
Wish I did.
Yeah, okay. I was about to really become friendly with you. But
Yeah, right.
Okay, major companies, 800-pound gorillas. These guys are the guys.
These are the guys, yeah, for global sterilization infrastructures. Yeah, Steris, they acquired Synergy, a European sterilizer, so they have a global footprint. And they acquired some interesting technologies like Cantel Medical, that's vaporized parasitic acid, formerly known as Revox. Steri-Tek purchased Nelson Labs, which is the gold standard of microbiology labs.
I know my friend Rob Packard has an intimate working relationship with them.
Oh yeah, Nelson Labs, that's like I said, the gold standard for microbiology labs. So these are very smart acquisitions. And Steri-Tek in 2015, they acquired Nordion, which is the sole distributor of Cobalt-60 for Gamma. So they cornered that market. Steris just announced, well, you know what, actually, I'm not gonna say that this is announced yet, because I don't see it, but they have they've made a bid at least for Mevex, which is the one of two e-beam X-ray accelerator manufacturers. The other
You should
being
go buy some stock right now.
Yeah, but that's privately held. So yeah, so nothing here is against any violations. This is all.
It wouldn't be inside information if you slipped and we all heard it and bought it. Go on.
Right. So yeah, so basically that's the setup. And then so you ask yourselves, well, who the heck are you, Steri-Tek? Well, Steri-Tek, we're basically, we are the innovator when it comes to e-beam and X-ray. And
what's your marketing copy?
Yes.
Go on.
That is it. And we have a long history. We started as NewTek way back in 1990 in conjunction with Stanford University to prove out e-beam for medical devices. And with NewTek, we had a long story, but we're in the San Francisco Bay Area where there is the Mass Rapid Transit is BART, Bay Area Rapid Transit, and we were right next to the BART tracks where our campus was, and they turned our facility into their maintenance facility. So through eminent domain, we had to close Steri-Tek down. It was shut down, and we had to start a brand new company, Steri-Tek, which was founded in 2016. But we're known for optimizing e-beam and X-ray for sensitive materials. We're the only e-beam facility in the world that's approved by the DEA to sterilize controlled substances, Schedule 2 and higher. And we have direct involvement. Our CEO is on AMI and ISO 11137 committees. Currently, we have our facility in the San Francisco Bay Area in Fremont, California, and right now we're building a brand new 100,000 square foot facility, like I mentioned, in the Fort Worth Dallas Metroplex at Louisville, which is north there, a little bit north of there, and we're going to be open third quarter of this year. We're going to have a grand opening, so if anybody is in the area, we would love to have you. You could connect with me via LinkedIn or whatever, and I would be making those announcements. And then we also have plans to build other facilities in the United States before 2023. So basically, within the terminal sterilization industry, now anybody that does terminal sterilization, you may see this, but there's big time capacity issues, whether it's ethylene oxide or gamma or E-beam. And there's different reasons why, which we will get into, but it's capacity issues. It's now we're at the point that you have to schedule time slots to get your product sterilized. And ethylene oxide, that is the go-to gas sterilization methodology. VHP, or vaporized hydrogen peroxide, is getting a lot more attention, particularly with the PPE issue that happened early on with the pandemic, with the shortage of PPE materials. There was the Battelle Memorial Institute, I don't know if you recall, but they developed a mobile system to sterilize PPE materials. But there is an AMI subcommittee developed, so there will eventually be a standard developed with that. Others, vaporized parasitic acid, nitrogen dioxide. There's a lot of interesting gas technologies, but it has a long way to go to displace ethylene oxide. And the reason being is that ethylene oxide is very, very good. It's very effective, and it's relatively cheap. So companies, for the last 50 years, really into the 2010s, really nothing changed in the terminal sterilization industry. Now with ethylene oxide, there's always been concerns because the residuals that are left on products are known carcinogens. That's been a long time problem. But what happened in basically starting like 2018, 2017 or so, big time issues arose with ethylene oxide because of the emissions into the environment. And even back in 1987, there was a study performed and a study released in 1989 in a plant owned by Steri-Tek, formerly Griffith Microsciences, that determined that this one plant emitted 170,000 pounds of ethylene oxide into the local environment. And that's just one ethylene oxide facility. So big, big problems there. And then really what happened now with that is with ethylene oxide, now there could be long and complex cycle times. It's a very arduous validation process. As I mentioned, it's a hazard to patients because of the known carcinogenic residuals. But the big issue really happened in the last few years. And it was centered around an ethylene oxide facility in Willowbrook, Illinois, just outside of Chicago. And it gained a lot of attention. And there was even a very sad story about a 45-year-old father that lived in the vicinity of this facility that developed stomach cancer. And even in Covington, Georgia, there was a study with Willowbrook that determined 6.4 out of every 1,000 people can expect to get cancer living in a 1.5 mile radius. So basically it became a big, big deal, and it still is a big deal. Just here in 2021, New Mexico Attorney General has announced another lawsuit against Steri-Tek for their emissions into the local environment. So there's been big time issues. The facility in Chicago has been closed down. There's a couple of facilities in ethylene oxide facilities in the Michigan area, Grand Rapids, that were closed down. One of the largest facilities in the world in Covington, Georgia, was closed down for a short period of time.
Jeff, does your company use ethylene oxide in any of your processes?
No, we do not use ethylene oxide at all.
And was that a conscious choice because of emissions or what?
Well, yeah. Well, actually, early on with Steri-Tek, our plans, we were going to have some small ethylene oxide units in our facility, which we did at NewTek, but it's especially in California. you have to develop specialized construction walls that will blow out in case of these systems, in case if there's an explosion, which there was in the early 90s in the Los Angeles area. An ethylene oxide facility exploded. And since then, there's been rigorous controls for construction of these facilities. But even at NewTek, it took us, we spent hundreds of thousands of dollars just on 3 units and have them installed. It took us 3 years to get it approved. And we were going to do that at Steri-Tek because ethylene oxide, there's some products you just have to use ethylene oxide. And also we were planning on having ethylene oxide to be able to help customers that have ethylene oxide sterilized products, but also to show them the light that the E-beam and X-ray is the way to go. But yeah, so
how arduous, you right, your first bullet point is it's the most widely used.
Yes,
And you
over
go on to tell us
55%.
about, you go on to tell us about all the reasons it sucks.
Well,
is there effort underway to convert people who are using this method to safer methods?
Well, yeah, companies, manufacturers are looking at it big time. Companies are scrambling to get out of ethylene oxide. However, it's not so easy. You
I
cannot
don't
just,
imagine it be easy.
yeah,
I imagine it be a hugely costly endeavor.
it's hugely costly because you have to for just strictly within the realm of sterilization validation, it's not that complex, but you have to redo all of your biocompatibility, shelf life testing, etc., leachables, extractables, etc. You have to redo that entirely.
As unlikely as it is, do you think it would require some kind of governmental regulatory legislation or tax incentive to invest in all of that to make the conversion?
Well, that's a great question, Joe. And indeed, the FDA back in 2019, which is not so long ago, but with this pandemic, it seems like a lifetime ago. But in 2019, the FDA, they issued a collaborative challenge to all medical device companies to identify new sterilization methods and technologies, number one. And then number two, how to reduce ethylene oxide emissions for existing ethylene oxide facilities. So that's afoot. So there is major initiatives here. And even for the cycle time, super concentrated ethylene oxide, they're looking at ways to minimize both the residuals, which is a much bigger deal in Europe than it is here, especially in France, but also with the reduction of ethylene oxide emissions. But bottom line is, yes, medical device companies are very knowledgeable with ethylene oxide, and they're looking to get out of ethylene oxide big time. And this has been happening actually for a few years. We work with a lot of
FDA
large.
challenge doesn't mean very much to me. If I'm a busy medical device executive with lots of other things to worry about, and I get a note that says FDA is challenging you, you're like, okay, well, that and me doing the New York Times Friday crossword puzzle when I get it.
Right. Totally true. Yeah. So, but basically, there was a Stryker, BD, and other companies are big participants in this, and they've already done studies. Yeah. So
Next
this
slide.
is okay. Excellent. So so basically, when we get down to irradiation sterilization, we're looking at gamma, E-beam, and X-ray. And they're all supported by ISO 11137, an international recognized consensus standard. And with the radiation sterilization, it's really, the result is the same. And it's the unit of dose is measured in kilogray. A kilogray is a kilogray, whether it's gamma, E-beam, or X-ray. So with gamma, basically, you're using radioactive material, cobalt-60, it's very reliable, highly effective for processing a wide variety of products. And it's the most widely formed of radiation sterilization used. And as we got into a little bit earlier, it's because it had an opportunity to take hold back in the 60s, because E-beam and X-ray technology at that time was not very good. And even going into the 90s, it was not that advanced. But now big time differences now. Now with gamma, it is more harsh to product than E-beam or X-ray. Basically with gamma, you have you have a grid array of cobalt-60. It's in a grid. They're cobalt-60 rods. They're in a grid configuration. It's kept underwater, under a chamber, and product fills that chamber. And when they're ready to sterilize, they bring this grid array up of the cobalt-60 rods, and that emits radioactivity. And the product absorbs that radioactivity over time in that chamber. So basically, a typical cycle is like 4 to 6 or 6 to 8 hours, depending on the strength of the cobalt 60, which is mathematically calculated. So gamma is very good, even though it's more harsh to product than E-beam or X-ray. But there's a big time issue with gamma, with the radiation, because there's a limited supply of cobalt 60. And Steri-Tek, they control, by purchasing Nordion, they control the cobalt 60 market, at least for the Northern Hemisphere. Steris has to purchase cobalt 60 from their arch rival, Steri-Tek, essentially. So there is a limited supply, and gamma facilities, they're jam-packed. So if anybody is doing gamma sterilization, you know what I'm talking about. Their turnaround time is now over 2 weeks. You have to schedule times to sterilize your product. Big time problems there. And in addition, with gamma, there's also issues with security and environmental issues. This is a very controlled, secure process for transporting cobalt 60. It's an amazing process, but there's issues and concerns, especially in other countries like South America, where terrorist groups could get a hold of this and create a dirty bomb. And also there's environmental issues because these nuclear rods, they lose their energy, which is again is mathematically calculated. And once that is depleted to a certain amount, these nuclear rods have to go into these very elaborate containers, and then they're buried, they go to a nuclear waste dump, which there's 24 nuclear waste sites in the United States, and they're all filled. So where are we gonna put this stuff? So we have to think about that too.
Okay.
Whereas, you were gonna say something?
I want to invite Mike Dolphin and Sue again, and maybe Andre, and anyone else who is working on a product that involves sterilization to type us a note and let us know what type of sterilization you are using. And if you've heard anything from Jeff that has you second guessing or wondering if you should be doing something different. Next slide.
Yeah, so we're glad to help in that area, and thanks for bringing that to everybody's attention, Joe. So we get into E-beam, and that, of course, is what Steri-Tek is about, E-beam and X-ray. Now, with E-beam, it delivers the dose very rapidly. The product is in front of it. With the E-beam, it's literally a beam of high-energy electrons, and the product goes on a conveyor, and it travels through an E-beam bunker, and I'll get a little bit for a demonstration to that, but product is basically in front of the beam for less than a minute, whereas with gamma, it's absorbing radioactive energy for periods of hours. There's a direct correlation between the time that the product is exposed to a source of radiation, regardless of the source, and the deleterious effects. So with E-beam, it's just less than a minute. So there's less time for oxidation to occur. And also with E-beam, especially when you get into drugs, biologics, and tissue product, and a tissue product, we're talking like allograft or xenograft tissue product, human tissue product, big time used or sterilized by E-beam. And it is the most efficient for low density medical devices, low density medical devices, packaging boxes versus gamma. Gamma is very good for more dense configurations. That's where you'd want to use gamma, and now in the future go into X-ray. Now
Mike,
also with,
I want to have Mike talk a bit because his company is new. He's yet to commercialize, and my house cleaner is here, so I must get up and let him in.
okay.
Mike, go ahead and tell us what you chose and why.
So my company, my current company is new, but we did a product before that with another company. With that company, we chose E-beam for all of the reasons that you've gone over, Jeff. You know, environmental concerns as well as just safety and everything. You know, we are here in Victoria, Canada. We're close to Vancouver, where Iotron is based. And, which has now been purchased, obviously. But so we went with Iotron, and they were great. They did a really good job. The only thing that we had to do, really, and this is of course super important for everybody, is to test the materials. So we had a range of different plastic materials that we ran through. We just got samples of it and ran it through, and it was amazing to see how they reacted to the radiation. A lot of them were discolored. A lot of them came out kind of yellow. And we ended up choosing the one material that didn't get discolored, and we were really happy with that.
Well, thanks for sharing that, Mike. And indeed, with the discoloration, that's due to oxidation. And there's different things you could do to reduce that, like with there's copolymers that keep the product clear, or turn it from a blue to a pleasing purple, for example. Modified atmospheric packaging, where you're reducing the amount of oxygen within the packaging environment, whether purging pouches with an inert gas such as nitrogen or argon. And there's also there's packets that are oxygen scavengers or free radical scavengers that you could include with the medical device packaging to reduce the oxygen environment. So there are things you could do to reduce that discoloration. And also that discoloration does remediate over time usually.
It does. It was actually really interesting to see, same day radiation was very yellow and almost brown, and then it would discolor or it would fade to a clear. We ended up going with a copolyester material that worked very, very well. It came out quite clear.
Good. Okay, well, glad to hear that success. And Iotron's a very good company. I know the individuals there. A lot of the eating technology really emerged out of Canada with Iotron and Mevax being the key players there. So then we get into X-ray. Now, X-ray is here. X-ray is the big time future. Now, why hasn't X-ray been around before? Well, it's been around for a while, but now the time has come. And the reason is basically with X-ray, you're using E-beam, electron beam accelerators. So you have high energy electrons that are emitted. Now with E-beam, you have the source, which is an industrial strength cathode tube. And then you have the gun, which is a vacuum tube that with sophisticated electronics to shape the beam. Then you'd have an electromechanical mechanism to cause the beam to go up and down to sweep in one direction, whether it's vertical or horizontal. And then you have the window that for the E-beam accelerator. Now with E-beam, that window or that source, that's titanium. And so you hit that window with high energy electrons, and on the other side, you're getting high energy electrons. And that's the mechanism of ionization. So basically with the radiation sterilization, you're breaking up the DNA and RNA of microbes, rendering products sterile. Whereas with gas sterilization, you're changing the size of amino acids, and amino acids enter into and out of cells. And by changing the size of those amino acids, when it exits the cell membrane, it basically bursts the cell membrane. So with gas, you're bursting cells, and with radiation, you're breaking up DNA and RNA. Now back to X-ray, where we're looking at with E-beam, we're using a titanium source or titanium window. You're hitting it with that with high energy electrons, which is the mechanism of ionization. So you're using high energy electrons. Now with X-ray, you change that window to tantalum. It could be gold, which is very expensive. We use tantalum. So basically with that window, you're hitting that window, you're still using the E-beam accelerator, hitting it with high energy electrons, but by changing this to tantalum, on the other side, you're getting photons. And in this case, photons is the mechanism of ionization, just as with gamma. With photons, you don't have mass, so it could go through much more dense materials. And that's the advantage of gamma, and that's the advantage of X-ray. But it's still, even when you come to X-ray and gamma, even though it's photons, it's the electron. You want to follow the electrons because it comes down to electrons. Even with photons, it's, I don't know how to pronounce it, but there's an effect that draws off electrons from molecules that cause the irradiation, even with gamma and X-ray. So with all of these, you come down to electrons causing the irradiation, causing the breaking up of DNA and RNA of microbes. Now, high-powered X-rays, it's the ideal alternative to gamma. It's environmentally friendly. You have the tightest dose uniformity ratio. You have higher penetration ability, even than gamma. And as with E-beam, it has flexibility to optimize the post-sterilized performance, including continuous temperature control, split dose processing, intelligent tooling. So there's a lot more you can do to optimize the post-sterilized performance of medical device products with E-beam and X-ray over gamma. And also with X-ray, as with E-beam, there's reduced material degradation as compared to gamma.
May I ask you to quickly just show us what the other four slides are in your deck?
Yes, so basically, this is a shot of our Fremont facility. It's basically a 10 million pound bunker. We have accelerators, 10 MeV 20 kilowatt accelerators, dual beam system. Boxes travel through the bunker, and then they encounter the first of two E-beam accelerators. They get E-beamed on one side, travel 10 feet further, and get E-beamed on the other side. Then the other slide, I was going to talk about material compatibility, drugs, and also important considerations. So
Okay,
for the sake of time.
No, we have the time. I brought Sue and Andre on. I specifically asked Sue about what choices she made, and Andre wrote me. And well, Andre, why don't you unmute yourself? Because he had a specific question for you, Jeff. Go ahead.
Oh, great. Hi,
Yeah, Jeff. Hi, Jeff. Nice to see you, and I appreciate the presentation. Actually, it's timely for us. We actually have a project for a client where we're developing a new endoscopic device that has a disposable, and the client has, in their design inputs to us, has specified ETO. And I'm just wondering, should we be, maybe as a service to them, recommending an alternative to the ETO?
Well, it's going to depend on what the material of that disposable is.
It's a combination of silicone and polycarbonate.
Okay. The silicone can be an issue. The polycarbonate will work out just beautifully. As Mike had mentioned, there may be discoloration that can be an issue. What we could arrange, Andre, is we'll do a complimentary E-beam run of this device, and you can evaluate it.
Okay, that'd be great.
Now, it might be that ethylene oxide is the best way to go, but you do want to,
define best though, Jeff, because you told us earlier about all of the detractions. So does best mean cheapest?
Well, no, because there's certain materials with ethylene oxide, and over 90%, certainly over 80% of all medical devices out there, components, disposables, what have you, can be equally sterilized by any of these methods.
And therefore, why wouldn't we go with one that's better for the environment and less expensive?
Right.
Very good question. Well, with ethylene oxide, there's certain type of products where that's the best terminal sterilization modality. Again,
define best. Are we talking? I mean, I don't know.
It's the best. It's the optimal choice. So if you have active electronics, if you have memory, a device that contains memory, integrated circuits, fluoropolymers such as Teflon, PTFE, you want to go with ethylene oxide. Yeah, so I mean, ethylene oxide, it's very important. There are detractions, there are negatives associated with it, but there are many instances where it is the best choice. And unfortunately, there's not better choices out there because what happened is, and this is for the whole medical device.
As a sterilization company, you've chosen not to even participate in part of the market that is the best and the most common. That's an interesting.
Oh yeah, big time. Yeah, because we see the writing on the wall. With ethylene oxide, companies are scrambling to get out of ethylene oxide as fast as possible.
And the most likely one to assume the throne then is what?
Well, E-beam and X-ray are rising to meet the challenge. But still, there's certain type of products where you still want to use ethylene oxide. Ethylene oxide is going to be here for a long time because basically all of these problems over the last 30, 40 years have been swept under the rug with emissions into the environment, patient safety. Because ethylene oxide is so cheap and it's so effective, medical device companies really didn't have a sterilization strategy early on. They just threw everything over the wall into ethylene oxide or gamma without giving forethought to the sterilization methods. And even with these issues that have happened in the last few years with the emissions that are known to kill people in the local environment, really the reason why this took hold in the 2010s in the last few years versus the 1990s is social media. This became a major Facebook issue, and in the community, seeing their loved ones dying because they live a mile away from an ethylene oxide plant. And so this spread like wildfire, and it took hold. So, ethylene oxide is very good. Now, if you live a mile away from an ethylene oxide facility, you're probably safe, but there are some big concerns there. But the real conflagration occurred with social media, and it's not letting go. And this occurred in the Trump administration, not to be political here, but we're likely with the new administration, we're going to see more scrutiny of environmental issues. So it's not good for ethylene oxide. Now, to be sure, the technology is always improving with the scrubbers, et cetera, and super concentrated cycles, but you still have emissions. But with that said, ethylene oxide is not going anywhere. But if your product is, you want to determine early on, if your product is sterile, is suitable to E-beam and X-ray, that's the way to go. Now, even with X-ray, Steris is getting big time into X-ray. They're building new facilities in Los Angeles, Chicago, and Boston. As I mentioned, Steri-Tek, we're building a brand new X-ray E-beam facility in Dallas. We're going to have another one on the East Coast very soon. So we're going to see a lot more X-ray here in the United States, and people will want to get away from gamma. So going from one radiation sterilization modality from another, like say from gamma to E-beam or gamma to X-ray, it's very, very smooth process. Whereas with ethylene oxide to radiation sterilization, it's you have to restart all over again.
Sue, would you tell us which, you wrote me a whole bunch of things and ISO things that went way over my head. What are you leaning toward for your processes?
Well, basically, I just copied and pasted the list of ISO certified sterilization techniques that we have to perform for a 510K approval. 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's like 12 or 13 different ones. And I'm not sure how we're going to do that, but you know, they all have different numbers and they all have different requirements.
Lucky for you, I know a guy.
Yeah.
And if you ever want any history on the history of the world of sterilization, this guy is like, I get it. You know who did what, when, I get it.
Well, also this is, you know, for my next startup, this is very important because it's gonna, I have another idea, but I can't talk about it. So
another one of the reasons I love this group so much is, like, I put sterilization on the agenda, and like most of the world is like, I'm not tuning in for that. Who cares? I don't do sterilization. And then there's like 2 or 3 people who are like, oh my God, this is the most timely you know, I'm so glad you did this today. So
it's timely for me because it's educating me. So thank you very much, Jeff. It's
my pleasure, Sue.
I'm really enjoying listening to you.
Oh, thank you. Yeah, so that's
us some more.
Okay, so yeah, so this is basically our configuration. With other E-beam facilities, you might have one accelerator, but we have a dual beam system. Very important to note, with other E-beam facilities, they have a tendency to go down, and they'll be down for weeks. With Steri-Tek, we're never down because we're basically our own backup supplier, having a dual beam system. So since 2018, we've been down 0 hours. Other E-beam facilities have had their problems. And also with Steri-Tek, we have walk-in freezers, walk-in refrigerators, the preconditioned product. So you want to look at advanced methods for sterilization. And with material compatibility, with the radiation sterilization, for the most part, the products are perfectly compatible to radiation sterilization. It's only, like I said, when you get into the fluoropolymers, Teflon, PTFE, where you could see some issues. What you have happen there with radiation is chain scissioning. You're breaking up molecules. In the case of fluoropolymers, you're basically pulverizing that with the radiation. So we have to watch out. So we want to determine early on, and we could certainly help with that to determine the most suitable sterilization modality. And Sue Galero is a real fine organization, and I'm sure that they have the expertise there as well to help you. And also,
what percentage of all medical device sterilization is done in ETL? Would you say more than half?
I would say, yeah, more than half, maybe 55%.
So are you turning away 50% of the people who go into your pipeline, your prospect pipeline? You have a conversation, you're like, yeah, well, you want ETL, that's not us. Goodbye, thank you, good luck.
No, we want to help. And we want to help. So even if a company comes to us and say, we got this new device and we've heard a lot of good things about E-beam and X-ray and Steri-Tek, we want to work with you. And then if we look at their device and whatever information they share, if we know that they're better off going to gamma, because X-ray is not quite here yet, or to ethylene oxide, we're gonna steer our customers and even inquiries in the right direction.
Of course.
So, yes.
Just I'm wondering about what percent of the
Oh, yeah.
conversations you have that end in, you know what, we can't help you because we don't do that modality.
Yeah. Well, there certainly with what had occurred with the Willowbrook facility in the Chicago area being shut down, companies were scrambling like crazy to. There was not a lot of forethought before this about sterilization strategy, because ethylene oxide is very good and very cheap. Gamma is very good and very cheap. So we've always done ethylene oxide. So we'll just do ethylene oxide for this next device. Companies did not think about the future and implications.
I'm curious, Mike, if you would unmute yourself, again, because you're a new entrepreneur and you're getting into this. When you started this business, what was your knowledge of sterilization technique?
Well, when we did our previous company, I had zero knowledge at all.
And how did you start? You know, you thought, oh, I need to get this sterilized. What did you do? Google sterilize?
We started doing a lot of research on different sterilization methods. We went through and came up with a lot of the same stuff that Jeff has talked about in terms of the benefits and costs and the concerns. And to be honest, the environmental concern of ethylene oxide was a big reason why we didn't use it. It's a big reason. And the fact that we had a local company here, Iotron, that was doing E-beam, we were like, let's try it. If it works, then that's going to be a really great option for us.
Did you make your choice based on, hey, they were local and they called us, or we filled in a form and we had a conversation, they sounded smart? I'm curious about how you fell into what you're doing.
We sought them out, and when we discovered that they were local, it added to it and it made it nicer. But truth of the matter is, we probably would have gone with E-beam anyway, just because of all the reasons that Justin talked about in terms of environmental concerns, safety concerns with gamma, the radiation, radioactive material, everything.
Research, is E-beam the most cost effective of the choices you could have made, or are you paying more for a benefit you perceive?
We might be paying more, but to be honest, the cost of sterilization is not a major factor in the cost of the device. You're looking at 10 or 15 cents a device for us, and that wasn't really moving the needle very much. If it was 5 cents, I wouldn't have cared. It would have been like, that doesn't make a big difference.
Is that a thing for you?
So again?
Moving the needle, is that a thing for your epidural device?
Yes, not moving the needle in that particular case. But yeah, so for us, it was, sometimes you pay a little bit more, but you're getting something that you're really comfortable with. And, one of the things we really liked about E-beam was that it was controllable. It's something you can dial up and dial down really easily. And if we wanted to do 22 kilograms or 25 kilograms, we could hit that exactly.
Okay. Jeff, if you would take us to slide 15.
I sure will. Well, this is the slide again. So, and also I want to bring this isn't really relevant to today's discussion.
Yep.
But we process a lot of drug products. That's very important because a lot of drugs are merging.
Well, it is insofar as their combinations, everything you're showing here. You don't sterilize something I put in my mouth.
No, well, you don't have to sterilize something you put in your mouth because you have so many germs in your gastrointestinal system. So
germs in your gastrointestinal system.
Yeah, I have plenty, I can assure you. Okay. Yes.
You have to sterilize the packaging in which my drugs go. Yeah.
Yeah, we sterilize everything. Yes. So whether it's a pre-filled syringe, whether it's an implantable that goes inside you, a drug-eluting.
If I take a bottle of aspirin, there's sterilization. When does it happen?
Oh, well, okay, sorry. Yeah, for aspirin, you don't have to sterilize that.
Okay, some other pill.
Orally. Yeah, you don't have to sterilize. If you're going to inject it, or if it's an implantable device that goes inside your body, then that has to be terminally sterilized. Gastrointestinally, you could eat dirt and you're going to be okay.
You eat dirt.
Yeah, have I ate dirt in the past? I sure have.
I bet. Okay, and next page.
All right, so again, very important, determine the optimal sterilization modality early on. And no, yes, and also with the EU and the FDA, EU, European Union more so, they have a mandate to exhaust all other sterilization modalities before resorting to ethylene oxide. Even major medical device companies that have their own ethylene oxide resources in-house, they're telling their design engineers, let's look at E-beam and X-ray before we resort to ethylene oxide or gamma. Big time changes. But you want to determine that optimal sterilization modality early on, determine the material compatibility and product functionality, which Mike had elaborated on and which Sue is going through this evaluation process with her very fine contract manufacturer, Galero, out of North Carolina. And also, as Mike had mentioned, with specialized techniques for E-beam, you could dial it in, and you could validate your product. Most medical devices for radiation sterilization are validated at 25 kilogray, meaning that they receive minimum of 25 kilogray. But if you have a very low bioburden, you could validate your product at even a lower dose. And Mike had mentioned 22.5. With MediMax, you could go down as low as 15 kilogray. With method 1 and method 2A, you could go as low as 8 kilogray. So there are, depending on how clean the product is, you could go to lower doses. And that is extremely critical for sensitive materials, to sterilize in as low and as tight of a dose range as possible. You also want to look at packaging and configuration. E-beam and X-ray are compatible with almost any type of packaging. Now for ethylene oxide, for example, you have to have breathable packaging. So for ethylene oxide, your devices are going to be in Tyvek pouches or Mylar, where it's breathable, where gas can enter. With irradiation sterilization, especially with E-beam, any type of packaging, thermoform plastic trays, Mylar, foil to foil, Tyvek, it could go through any packaging. And you also want to look at modified atmosphere packaging to further optimize post-sterilized performance. And you also want to consider what's going on within the contract sterilization industry itself. With Steris and Sterigenics, great companies, but they are built for the major medical device companies. You're not going to get a lot of collaborative effort in working with them, whereas working with Steri-Tek, we have a fine team under one roof. We could provide turnkey sterilization validation services, consulting on packaging, configuration, optimizing all aspects of sterilization.
And you get Jeff Sauter himself.
And then you have me, right? You can't, hopefully you can't beat that. So, and also consider the sustainability for the future. So, and it has been even until recent years, you know, we all knew about the problems with ethylene oxide. We all knew the problems with gamma, but really nobody spoke up because they're so darn good and they're cost effective. But now things are changing. So the time is now for E-beam and X-ray. And with that, that's finally the end of the presentation. I totally went over time.
Thank you.
So that's what we're about. I hope this was good for everybody. Any questions?
I've been feeding them to you as they came. It's,
okay, excellent.
Like I said earlier, it was a niche presentation where I'll confess attendance was a little lower, but not everyone needs to sterilize anything. But
right.
People who do really needed to hear what you had to say.
Yes. Yeah.
No, it's not quantity, it's quality, and this is important. This is important especially for me for development purposes. I could see a lot of contact between myself and Jeff.
Great.
Jeff, to that effect, I know you're not a frequent logger onto the platform. Maybe for a week, you know, let the excitement of the Sauter presentation die down a bit. Log in every day. Let these kind folks have an easy way to communicate with you. Less
certainly, yeah,
relegated to the archaic old forms of email and phone. I mean,
sure. Yeah. So I'm available. Please reach out. Let's see. Do I have my phone number on the last slide?
What you have is Jeff's on premium, and you can write him there.
Yeah, just connect with me via Slack. And Joe, I am improving on that.
You are. It's glacial, but you know what?
It's glacial, yes.
We have some new behaviors here. Oh, next week I'm excited about. We are going to be talking about empowering women regarding breast cancer monitoring. So I'm definitely going to get our friend Shabir back on the phone. You'll recall a few weeks back he talked about his breast cancer detection product. So kind of coming all around. And I look at next week's as one of those concentrated consulting kind of efforts where she's not yet commercialized, and collectively we get a few smart people in the room to hear what she has to say at once, and maybe we can give her some direction and accelerate where she's going. Mike and Sue, if and when you want to avail yourselves to the same, that's kind of what we're here for. So on behalf of Joe Hage and the Joe Hage family, this is Joe Hage signing off for this week. I love these. Thanks, everybody, and see you soon. You know where to find me.
Thanks, everybody. Thanks,
Thank you,
Joe.
Jeff.
Okay, take care, everybody.
Bye.
Bye.
Thank
Bye-bye.
you, Jeff.
Thank you. Bye-bye.
Thank you, Jeff.